Bismillahirrahmanirrahim, and welcome back to this next video. And this is the continuation of the previous video where we were discussing the differences between the prokaryotic and the eukaryotic cell. Now, in this particular video, I'll continue the previous discussion and I'll be giving you some of the differences between the prokaryotic and the eukaryotic cell. And I'll be focusing on why this is important, why we study the differences between the prokaryotic and the eukaryotic cell. So, let us talk about the first feature, the nucleus. When we talk about the prokaryotic cell, there is no true nucleus, and the DNA that is present in a particular region in a prokaryotic cell, which is known as the nucleoid. When you talk about the eukaryotic cell, they have got a true nucleus. By that I mean that they have got a proper nuclear membrane which is surrounding the genetic material. If you look at this particular image, if this is a prokaryotic cell, this is the nucleoid region where the genetic material that is present, but that is not surrounded by any nuclear membrane. And you can see a very clear nuclear membrane in the eukaryotic cell which is surrounding the genetic material. If you talk about the size between the prokaryotic and the eukaryotic cell, the prokaryotic cell they are generally smaller, and the eukaryotic cell they are generally larger than the prokaryotic cells. And these are actually the ranges where you can see the size of the prokaryotic cell. They can be from zero point 0.1 to 5 micron. The eukaryotes they are from 5 to 100 micron. Another important difference between the two is that if you talk about the cell structure and the organelles, so in prokaryotes, the cell structure that is very simple and they have got a few organelles. In comparison to the prokaryotic cell, the eukaryotic cell have got a complex cell structure and they have got numerous organelles in comparison to the prokaryotic cell. Looking at this particular image, if you see over here, you have got the ribosomes in the prokaryotes as well as in the eukaryotes. But if you focus on this particular diagram of the eukaryotic cell, they have got other organelles like the Golgi apparatus, the endoplasmic reticulum, both the smooth one and the rough one. So the organelles, their number that is higher in the eukaryotes in comparison to the prokaryotes, and the cell structure of the eukaryotic cell that is more complex in comparison to the prokaryotic cell. Another important difference is when you talk about the DNA or the genetic material. So the prokaryote that actually have a circular DNA without the famous histone proteins. But when you talk about the eukaryotic cell, you have got the linear DNA, but they've got these histones over here. If you look at this one, if this is the DNA of the prokaryotic cell, you do not see any histones over here. But if you look at the eukaryotic DNA, this is a linear one, but that particular DNA is wrapping around these histone proteins, and this wrapping around these histone proteins is actually giving a compact structure to the DNA of the eukaryotic cell, thereby helping its accumulation in the nucleus of the cell. And we will have a detailed discussion on that when we talk about the nucleic acids, about the DNA and its structure. Another important difference between the two is if you look at the cell division or the reproduction in the prokaryotic cell, so they usually divide by the process of the binary fission, and the mode of reproduction in prokaryote is asexual. When you talk about the eukaryotic cell, they divide by the process of the mitosis. If you talk about the somatic cells, and they divide by the process of the meiosis. When you talk about the germ cells like the sperms and the eggs. So this is a diagrammatic representation of the binary fission. There is a parental cell, it duplicates its DNA, cytoplasm divide, and you have got two daughter cells with the same genetic makeup is the parent cell. When you talk about the mitosis, the same thing that is happening over here. You start with the 2n and you divide a single parent cell into two daughter cell, again with the genetic makeup of 2n. In meiosis, you start with the 2n, but in the end, you are going to get 4 haploid daughter cells, and all of them they have got 1n genetic makeup. That means you have decreased the genetic material by half. Another important difference between the two is that is the ribosome, because we'll be talking about the translation in detail during this particular course, and the translation between the prokaryotic cells and the eukaryotic cell. They've got some differences which we will see over there. But this particular time, just keep in mind that the ribosome of the prokaryotic cell, that is smaller, that is 70s ribosome. The ribosome of the eukaryotic cell, that is a larger one, and that is 80s. Now, if you look at this one, if this is a 70s prokaryotic ribosome, they have got one large subunit of the, which is known as the 50s subunit. They've got a small subunit which is known as the 30s subunit. There are two types of the RNA in the large subunit. One is known as the 5s RNA. The other one is known as the 23s RNA. In the small subunit, they have got the 16s RNA. If you talk about eukaryotic ribosome, they've got two subunits again, a large subunit which is 60s subunit, and they've got a small subunit which is the 40s subunit. And in the large subunit of the eukaryotic ribosome, you've got three types of the RNA. One is the 5s RNA, another one is 5.8s RNA, and then you have got the 28s RNA. And in the small subunit, which is the 40s subunit, you have got the 18s ribosomal RNA. So what this S mean? This S is actually the Schwarzberg unit, which is a non-metric unit for the sedimentation rate. And based on the Schwarzberg unit, you actually differentiate between the prokaryotic ribosome and the eukaryotic ribosome. Another important difference between the two is that the prokaryotic cell, they have got a very rudimentary type of the skeleton, or the skeleton is almost absent. But the eukaryotic cell, they have got a well developed cytoskeleton. So the cytoskeleton is actually the protein complex, I would say, or the combination of the protein that give particular shape to the cell. So if you look at the eukaryotic cell, they have got these actin filaments over here, they've got these microtubules over here, and they have got these intermediate filaments. And these three protein filaments, they are actually responsible for the formation of the eukaryotic cytoskeleton. This one is a better image of the microtubules and the intermediate filaments and the actin filaments, and collectively these three types of the proteins, they are making the eukaryotic cytoskeleton. So the cytoskeleton is very prominent in the eukaryotic cell, that is very weak, or you can say that is actually absent in the prokaryotic cell. So why are we studying the differences? This would be the main question in your mind, that what is the advantage of studying the differences between the prokaryotic and the eukaryotic cells? Let me give you some of the examples. Studying the differences is very important in the development of the antibiotics, and hence in the control of the infections. For example, if you talk about the cell wall inhibitors, the penicillin that inhibit the synthesis of the peptidoglycan. This peptidoglycan is a very important component of the bacterial cell wall without affecting the human cells. So whenever you talk about a drug, the most important target for the drug is that that particular target is not present in the host, and that is present in the pathogen which is responsible for causing the disease. So the penicillins, they are inhibiting the synthesis of the peptidoglycan. So is the peptidoglycan that is not present in the human cells. So this penicillin that's only targeting the pathogenic bacteria and not the human cells. So this is one of the importance, that if you look at the differences between the two, you are going to identify certain targets which you can exploit for the treatment of that particular condition. Secondly, if you talk about the protein synthesis inhibitors, another class of the antibiotics like the erythromycin, the tetracycline, the chloramphenicol, and the aminoglycosides, what these protein synthesis inhibitor antibiotics do, that they selectively interact with the 70s bacterial ribosome, and they do not interact with the 80s eukaryotic ribosome particle. What that would mean is that they are only going to inhibit the protein synthesis in the bacteria because the target is there, and they are not going to stop the protein synthesis in the humans, for example, because the 70s ribosome is not there. Another important application, I would say, is the biotechnology and genetic engineering, because understanding the prokaryotic genetics, that is going to facilitate the use of bacteria in genetic engineering. For example, Escherichia coli, most famous prokaryote that is commonly used in the recombinant DNA technology to produce insulin and other important protein. So once you have known the molecular biology of the E. coli, you can use that for the purpose of the benefit of the human being. In this particular case, the production of the insulin. The molecular biology is also very important in the cancer research. As I've told you, that drug targeting, that is very important, and identifying unique features of eukaryotic cells that allow for the development of targeted cancer therapies that minimize damage to the healthy cells. So these applications that I'm giving you, that is, you can see, focusing on two of the aspects, that is giving you the importance of studying the differences between the prokaryotic and the eukaryotic cells, and they are also giving you the clue why studying molecular biology is important. CRISPR technology, this CRISPR technology that is derived from the bacterial immune system, and we are using the CRISPR-Cas9 technology in the experiments in the labs these days for the precise editing of the eukaryotic genome, leading to advances in the gene therapy and genetic research. And this gene therapy, that would be the future for treating the genetic disorders that we are unable to treat in the current era. So we are going to continue the discussion in the next video.